Hello, welcome to Biology, the scientific study of life. Again, more about endocytosis. Bulk transport across the plasma membrane occurs by exocytosis and endocytosis. Water and small solutes enter and leave the cell by passing through the lipid bilayer of the plasma membrane or by being bumped or carrying across the membrane by transport proteins. However, large molecules such as proteins and polysaccharides, as well as larger particles, generally cross the membrane by a different mechanism involving vesicles. Exocytosis. As we described it, the cell secretes macromolecules by the fusion of the vesicle with the plasma membrane. This is called exocytosis. A transport vesicle that has binded from the Golgi apparatus moves along microtubules of the cytoskeleton to the plasma membrane. When the vesicle membrane and plasma membrane come into contact, the lipid molecules of the two bilayers rearrange themselves so that the two membranes fuse. The contents of the vesicle then spill to the outside of the cell, and the vesicle membrane becomes part of the plasma membrane. Many secretory cells use exocytosis to export their products. For example, certain cells in the pancreas manufacture the hormone insulin and secrete it into the blood by exocytosis. Another example is the neuron or nerve cell, which uses exocytosis to release neurotransmitters that signal other neurons or muscle cells. When plant cells are making walls, exocytosis delivers proteins and certain carbohydrates from Golgi vesicles to the outside of the cell. Endocytosis. In endocytosis, the cell takes in micromolecules and particular matter by forming new vesicles from the plasma membrane. Although the proteins involved in the process are different, the events of endocytosis look like the reverse of exocytosis. A small area of the plasma membrane sinks inward to form a pocket. As the pocket deepens, impinges in forming a vesicle containing material that had been outside the cell. There are three types of endocytosis: phagocytosis, which called also cellular eating, and penicytosis, cellular drinking, and respiratory mediated endocytosis. Human cells use respiratory mediated endocytosis to take in cholesterol for use in the synthesis of membranes and as a precursor for the synthesis of other steroids. Cholesterol travels in the blood in particles called low density lipoproteins, LDLs, complexes of lipids and proteins. These particles act as ligand, general term for any molecule that binds specifically to a receptor site of another molecules. By binding to LDL receptors on membranes and then entering the cells by endocytosis. In humans with the familial hypercholesterolemia, inherited disease characterized by a very high level of cholesterol in the blood. The LDL receptor proteins are defective or missing, and the LDL particles cannot enter cells. Instead, cholesterol accumulates in the blood, where it contributes to early atherosclerosis, the buildup of lipid deposits within the walls of blood vessels, causing them to bulge inward and impede blood flow. Vesicles not only transport substances between the cell and its surroundings, but also provide a mechanism for rejuvenate or remodeling the plasma membrane. Endocytosis and exocytosis occur quantitatively to some extent in some eukaryotic cells, and yet the amount of plasma membrane in a non-growing cell remains fairly constant over the long run. Apparently, the addition of membrane by one process offsets the loss of membrane by the other. Energy and cellular work have figured prominently in our study membrane. Notice this one. Endocytosis in animal cells. Notice here the picture. In phagocytes, a cell, notice this one, a cell engulfs a particles by rubbing um, notice that pseudopodia around it and packaging it within a membrane enclosed sac large enough to be classified as a vacuole. The particle of is digested after the vacuole fuses with a lysosome containing hydrolytic enzymes. Notice. This food or other particle, and this one is pseudopodia. Notice this closer look of this one. Pseudopodia of amoeba, bacterium, and food vacuole. An amoeba, an amoeba engulfing a bacterium phagocytosis. Another picture, penicytosis. Notice plasma membrane, vesicle, and bone. In penicytosis, the cell clumps droplets of extracellular fluid into tiny vesicles. It is not the fluid itself that is needed by the cell, but the molecules dissolved in the droplet, because any and all included solutes are taken into the cell. Penicytosis is non-specific in the substances it transports. Notice that vesicle. Notice penicytosis, vesicle forming arouse in a cell lining a small blood vessel. Notice that. Notice also the size, 0.5 micrometer. Respiratory mediated endocytosis. Notice coat protein, coated vesicle, coated pit. Respiratory mediated endocytosis enable the cell to acquire bulk quantities of specific substances, even though those substances may not be very concentrated in the extracellular fluid. Embedded in the membrane are proteins with the specific receptor sites exposed to the extracellular fluid. The receptor proteins are usually already clustered in regions of the membrane called coated pits, which are lined to their cytoplasmic side by a fuzzy layer of coat proteins. Notice this. Extracellular also substances ligands bind to these receptors. When binding occurs, the coated pits forms a vesicle containing this one. The ligands molecules. Notice that there are relatively more bound molecules parable. Notice this one, parable inside the vesicle, but other molecules green one also present after these ingested materials is liberated from the vesicle. The receptors are recycled to the plasma membrane by the same. Thanks for listening.